Hi, we're doing a journal club again. I am Mike. I will be your host for today. I am from T-Lab Monash, first year PhD student. Vishaal.
Hi, yeah, my name is Vishaal. I am a master student at T-Lab as well.
Hi, I'm Lipeng. I'm currently the research assistant in Lab Monash as well.
All right. So today we are doing the paper, Multi-Model MRI Reviews Brainstem Connection that Sustains Wakefulness in Human Consciousness. First, well, it's a very technical paper, but first, the author divided give a sort of operational definition of consciousness. They believe there are two parts to consciousness. One is awareness, the other is wakefulness. For example, a person in a vegetative state may be wakeful, they are awake, their eyes open, but in fact, they are not aware of anything at all. Um, and the author argued that we have actually learned a lot about awareness in the recent decades, especially this default mode network that seems to be responsible for a lot of our self-awareness, introspection, and so on and so forth. Now, the default mode network is mainly located in the cerebral cortex. It includes areas such as posterior medial complex or medial prefrontal cortex. But we actually know very little about the other half of consciousness, namely the wakefulness part, the physiological arousal. The author want to argue that there is something similar to the default mode network, but for the arousal. They call it default ascending arousal. Now, it turns out that we don't know a lot about this, largely because the nuclei of this network is a lot of it is located in the brainstems. And it's very difficult for conventional imaging methods such as fMRI or EEG to have the spatial resolution required to investigate the subcortical nuclei. Not to say that no one has done it before, though. Some studies have shown that the reticular formation seems to play a role in wakefulness. When a person emerges from their sleep and become awake, there seems to be increasing metabolic activities in the reticular formation. But that's sort of all we know about this network. So the author decided that they want to do things a bit different. They want to investigate the structural connectivity of this network using these ex vivo methods, using the preserved brain of people who have passed away. Now, how are they going to do it? Well, they first gathered up three brains. Um, and first of all, they need to find the starting point of this network, this ascending pathways. So where are they? Well, they are in the brainstems. But where exactly in the brainstems? The author were able to identify 18 potential candidates of these arousal nuclei in the brainstems based on either the data collected from previous studies, general anatomical knowledge, and so on and so forth. And then they used this diffusion-weighted MRI as well as immunohistochemistry, staining the brain, to track the pathway from these 18 nuclei upwards and see uh, what are their connections. Interestingly, for almost all 18 candidates, they have some sort of connections in the upper levels of the brain, either in thalamus, hypothalamus, basal forebrain, whatever it is, there are some connections. And some of them, for example, if I may show my screen for a brief moment. Now, give me a second. Now, hopefully, there wouldn't be any copyright issue since I'm only showing it for a brief moment. If we're going to see the figure C, this is actually the pathway from the medial reticular formation to the other areas of the brain. Now, as mentioned before, reticular formation seems to play an important role in wakefulness, and indeed, they did found this very clear pathway upwards, which is great. Now, another very interesting interesting questions they want to tackle is how is this default ascending arousal network related to the default mode network? So how does the awareness part related to the wakefulness part in the brain? So they conducted these sort of analysis based on both the probabilistic analysis and deterministic analysis using the diffusion MRI. And they did find actually very extensive connection between DMN and their newly funded DAAN. It's best to show with another diagram. Give me a quick second. So this, if we can look at figure 3, we can see that as previous study has demonstrated, the reticular nuclei, as well as the nuclei outside of the reticular formation, does have very good connection with both PMC and MPFCs, which are the two main parts of, well, yes, two very important part of the default mode network. And now that they found these interesting structural connectivities, they actually also interested in whether the structural connectivity can be matched up with functional connectivities. So they collected data from the Human Connectome project, and they compared and contrast, trying to find a correlation between the two. Unfortunately, it seems that for most of the 18 candidates, the correlation isn't great between the functional connectivity and the structural connectivities, as we can see in the figure B here. Most of them either have good connectivities but bad structural connectivity, good functional connectivities but bad structural connectivities, or they have reasonably good structural connectivities but bad functional connectivities. This is with the exception, perhaps, of the VTA, which is the ventral tegmentum areas, an area predominantly based on the dopamine neurotransmitters. It seems this area may play quite an important role in connecting the wakefulness part of the brain to the arousal part of, the wakefulness part of the brain to the awareness part of the brain. And the recent studies on people who have suffered brain injuries seem to suggest something quite similar. So that's an interesting direction for future studies. At the end, the author also mentioned a few limitations of the diffusion MRI, some of the problem that it may face. For example, it doesn't really tell you which direction the signal is sending towards. The author are imagining a ascending pathway from the midbrain upwards, but it's possible that it's actually a descending pathway. There are other sort of limitations. For example, they only have 3 brains, which, it's reasonable number for an ex vivo study, but, it's still only 3 brains. So yeah, that's roughly it. That's the rough idea of the article. And have I missed anything? I'm not very familiar with all the technical aspect of the paper. So please do mention it if I have missed something.
I think that's pretty good. One thing I want to highlight is that this kind of paper is research sources paper, not kind of article. So the main object of this research resource paper is designed to make the study data and the methodology accessible to the other scholars, usable for future study, advancing their like scientific knowledge in this spaces.
Yeah.
Any questions then related to the paper itself?
Yeah, given the nature of this research resources paper, so I am thinking about how we can use their data to study consciousness. But you know, we are focusing on the content of consciousness, but this paper is mainly focused on the consciousness state, right? The wakefulness, and then the wakefulness versus, for example, the coma vegetative state. So what we can do, I'm thinking about this question. Do you have any idea?
I actually think maybe Vishal can answer this for us, but I think it could be quite interesting to see how it would work in dreams, right? In dreams, you have a state where the person is not wakeful, they're actually sleeping. Perhaps the physiological arousal will be quite low, but they are aware in a sense. So in that case, we could see something very interesting about how the two network interact with each
Yeah, especially because the authors did make that distinction, right? Arousal is basically physical of the activation of the brain instead of wakefulness, and awareness is basically content of consciousness, right? So yeah, you're absolutely right. I think looking at the distinction, given those two definitions, right, it's particularly it will be useful to see, or be very interesting to see in the context of dreams for sure, right? Um, I believe the reticular activating system is also quite involved in sleep and dreams. So there's also a pretty deep subcortical structure, right? So um something that the authors also mentioned, that the one of the challenges in or one of the reasons why subcortical networks have not really been studied all that much is because of um challenge in having tools with good spatial resolution. So yeah, I mean, this is, I guess, is the first step. I guess, but I think just to clarify as well, right? Like the 13 nodes that are proposed in this ascending network, they're all derived from previous human and animal studies, right?
I
Right,
believe so.
basically.
Yes.
Yeah. Okay. So this is basically a proposed network. It's not really an established like causal, like there's no data to show that, this is actually um indeed a network that is involved in sustained wakefulness. But this is definitely a venue for future exploration. Um yeah. But what stood out to me the most is the one node in particular, the ventral tegmental area, because before this you wouldn't really think about that area in terms of consciousness, right? Because it's more to do with um I guess learning, cognition, the reward system, and so on and so forth. So I think that's a pretty new pickup. Yeah. But yeah, I apologize if I'm deviating like so quickly from the original question, but I just find they're all very pretty interconnected. So yeah. Yeah.
Yeah, I kind of wonder whether, I mean, as a dream is, of course, an example, whether, conscious experience can occur without wakefulness. But I suppose also are more focused on the therapeutical application of these, and coma and vegetative states are what they are really concerned about. And I suppose in that case, there's a clear separation. But for
Yeah.
us, I feel like the separation isn't so obvious. The wakefulness, I'm uncertain how important it really is for consciousness.
Mm. Yeah. Um, yeah, I don't know. It will be it would be great if you could confirm that this network, right, in terms of the two aspects, uh, wakefulness and sorry, arousal and awareness, and see which one of these nodes are actually most involved in one or the other, or even both. So yeah, but I guess it's also highlighted in the limitations that it can be a bit difficult to assess this. Yeah.
Yeah, especially if we look at the correlation between the functional connectivity and the structural connectivity. I'm kind of not sure how to feel about that because well, the study have used all these seems pretty advanced methods to really tackle the structural connectivity part of the question. But it's kind of disappointing that there is just very limited sort of correlation between the two, which worries me a little bit. Perhaps it's just we are still at the beginning stage. Maybe that's the reason why. But it doesn't feel good, does it? Just look at the correlation.
Well, I think that figure 6 you are talking about is a more, I think the more important part is the pattern. The pattern is very similar, and right. So you
Uh,
can see figure 6b and c. The pattern is pretty similar, but yes, as you said, the absolute value is pretty small. But I think the pattern is more important. I'm not sure.
Uh, can I clarify what do you mean by the pattern?
Pattern, you can see VTA, the position of VTA related to the others is very similar to dopamine, related to to serotonin and glutamate and acetylcholine.
Oh yes, 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 okay, yeah.
uh,
Yeah. But
let's
wait, I think
Maybe I have misinterpreted this.
yeah, to my understanding, the figure 6C basically shows the predominant neurotransmitter of the cluster of nodes. Yeah.
Ah, I
Yeah,
see.
yeah. So basically, because for example, the ventral tegmental area, the VTA, again, like I mentioned, okay, it's involved with the reward system, reward circuitry and cognition. It has a lot of dopaminergic circuits, right? So I think in that case, for example, in where the VTA is, it shows that the predominant neurotransmitter is dopamine.
Okay, get it.
Yeah.
Mm.
out of curiosity about this graph, so if you look at the dopamine and you look at the VTA in both graphs,
Yeah.
they are at the exact same spot, right? In terms of functional connectivity and structural connectivity.
Mhm.
So does that mean VTA was the only dopamine like predominant uh, nuclei that they have?
Mm. Can we
Kind of.
Sorry, could I clarify
So
what do you mean again?
so if you look at the location of both
Yeah.
the VTA dot and dopamine dot, they are at the
Mm.
exact same location.
Mm.
0.05 function connectivity and just below 0.02 structural connectivity. It seems to
Yeah.
me that there is only one nuclei that's predominant by the dopamine neurotransmitter.
Um,
Or can we say VTA, the functional connectivity of VTA and the structural connectivity of VTA is the same as or the similar to dopamine? I don't
But so the dopamine if my understanding is correct, the C represent each clusters of nuclei, right? So in
Mm,
some cluster there may be five different nuclei, let's say all belong to the glutamate dot in the figure C. Maybe three
yeah.
belongs to the serotonin, whatever.
Yeah.
But my question is whether there is only one belonging to dopamine, and then that is a VTA.
Uh, I don't think so.
No?
I think yeah, I don't think so.
Hmm.
I don't know.
Maybe just a coincidence there.
Yeah, I'm not sure. Wait, let me just check this real quick.
Because I was going to say that if assuming that VTA was the only dopamine nuclei that they have, it's sort of difficult to say whether dopamine really plays an important role, or is it
Mhm.
just VTA areas in some way special?
Right. Well, I might have misunderstood figure 6C actually, but
Oh,
yeah. Um, the brain's increment. I mean, yeah. So my understanding is like the nodes in figure 6C are basically the neurotransmitter type, the predominant neurotransmitter type of each of the nodes. They're just represented in a cluster. Like for example, PAG, PDG, and MRT, the predominant the predominant neurotransmitter is glutamate, for example.
yeah, yeah,
Yeah,
yeah.
Okay.
but yeah, but
Ah,
yes.
okay. Either way, we can probably
But
move
yes,
on.
okay. I mean, we're diving too much into the technical
Yeah,
detail, it seems like. Yeah, so
yes.
yeah, okay.
Um, hmm.
But yeah, any other thoughts, I guess, on this proposed network?
I have a thought
Yeah.
regarding concern to figure four. For
Mm,
example, figure four giving giving the connection uh, between nodes and giving the connection like intensity or the intensity of this connection.
mm,
So basically, we can build a structure between this node, I guess, based on this relationship and the nodes. For example, if we use if we convert connectivity probability into the distance, if that's the case, we can build a structure of such kind of structure of
but isn't this parallelogram generated from the data of the mapped structure connectivity?
sorry, what do you mean?
Um, Sorry, isn't this parallelogram based on the mapped uh network, subcortical network that they highlighted? That
Mhm.
is essentially the structure, right? That you're talking about? Or like
Oh,
when you
I
say,
mean doing the like a 3D structure like
oh,
that,
okay,
you mean?
okay.
Yeah. To having
Uh,
there the data of connectivity, the data of nodes, it is easy to build a similarity metric, right?
okay, okay.
I suppose the real question is why are we building this network? Would be the question.
Mm-hmm.
And then we can have this structure maybe serve as the baseline for us to investigate another conscious phenomenon.
But why? Why
Mm.
would we think the two?
For example, if we are investigating dream, if we use dream to compare with this baseline structure, I think maybe we can find something interesting.
Okay.
Okay, it just some I don't
It,
know.
it's possible, right? It's possible that in some way certain experience reflect some sort of structure in the physical brain. It would be
Mm.
interesting if that's the case, although it feels somewhat unlikely.
It is the case. We called it isomorphism between the phenomenal consciousness and the brain activity from one domain to the other domain. For example, if we have a phenomenal consciousness structure, which is the qualia structure we are investigating, then we might find a isomorphism, which is the brain activity structure correspond to the phenomenal.
Okay. Yeah.
I think this is the second step of our qualia structure project.
I see. You're basically comparing structures between two.
Between neural activity and the phenomenon.
Yeah,
Um,
yeah, yeah. Okay. Yeah, yeah.
my
Yes, yes,
worry
yes, yes.
is that it's not exactly a neural connectivity graph. It's
Yeah.
a structural connectivity.
Uh,
And I doubt that actually, maybe this is unkind of me, but
mhm.
I'm thinking that if you have the structure of this, sorry, well, yeah, the structure of this structural connectivity graph, and if you have the structure of a functional connectivity graph, I'm not even sure that they can align with each other very well at the moment. Maybe in the
Mm.
future it will change. But
Given figure 6b, right? Yeah, yeah.
yes, exactly,
Yes,
exactly.
yes, 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 yes.
Ah, I get it. I get it. So I'm not talking about really alignment. There must be a function that transfer one domain to the other domain. I'm trying to find this function. Right.
Mm.
Anyway, that's that's out of this
Yeah.
discussion.
Okay.
Very interesting though. Hmm.
Mhm.
Mm. But I believe they did make some form of comparison, right? With the mapping this ascending network with, I guess, the MRI data from the Human Connectome project. What was the
That's
wait,
the structural and functional connectivity part, I
right?
believe.
Yeah, 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 yeah. So in a sense, they did make some form of comparison in that manner.
Ah, yes.
Yeah, but and that revealed that it's not exactly one to one. So yeah. Well, except for the VTA, which is pretty interesting, I have to say, right?
Can you tell us more about the VTA? Because I don't think either Lipeng or I are very knowledgeable about the
I
area.
mean, so I don't know much, but from my prior, I guess, learning of somewhat a little bit of the topic, of neuroanatomy. Um, yeah. So the midbrain is basically the mid part of the midbrain is the tegmentum and the tectum.
Yeah.
Okay. And the VTA is, of course, is part of the tegmentum, and it basically has a lot of dopaminergic cell bodies.
Mm.
All right. And so it's part of a specific dopamine system, part of it, and dopamine pathways, but I forgot what name of the pathway is exactly, but it essentially plays a role, most notably in like your reward system, basically,
Mhm.
right? So your reward circuits, right? Motivation, um, yeah, associative learning and so on. So
Okay.
yeah. So, but I think yes, more recent studies have also showed that I guess it plays a role in arousal, but yeah, that's not the I don't think that's the conventional like function
Yeah.
that was found for the VTA, but it more recently is found in arousal. So I think yeah, this the fact that this paper highlights the VTA is quite a notable node. as among the 13 nodes of this active ascending activation ascending arousal network. Sorry, yes, resonant or not network. It's pretty interesting. Yeah. So,
Hmm.
yeah.
Yeah, when we are talking about wakefulness or sleep, dopamine is, at least as far as I know, it's not the first hormone that you think about.
Mm.
But this I may actually reveal that dopamine have a very specific impact on the physiological arousal.
Mhm.
Hmm.
Yeah.
Any other question?
Mhm. Not really from me, no.
Uh,
I think it's a pretty good network. I mean, this default AN. I think it's a very interesting um the neurophysiological I guess correlate of consciousness or sustained wakefulness, right? But it would be great if we can test how involved each of these nodes are in basically sustained wakefulness, yeah, arousal or um
hmm.
awareness, yeah.
Yeah, I assume they have some lesion data from previous study to back it up, but it would be interesting to see a more targeted study. I agree.
To be honest, maybe I only get 40 or 50% the content of this paper. So regarding figure 5, I'm not quite understand how to interpret C and D. Do you have any can you help me with that? I don't.
Uh, I, to be honest, don't understand figure five very much either, which is why I'm
Yeah, I
trying
mean,
to not talk about it.
yeah, there's quite a bit of technicality in some of the discussion here, so yeah,
Okay, I
yeah.
see.
Let's see. Yeah.
Yeah, I think that's mostly from me as well. Maybe the last comment from me would be that the ex vivo study is actually something I never thought about before. I didn't know you can do diffusion fMRI, sorry, diffusion MRI on, preserved brain rather than living ones.
Mhm.
That's a very interesting aspect for me.
Oh, so yes, I just searched for some Wikipedia and Google. They are saying that they're looking at this technique. Very interesting. Can do it on separate the brain and then because in my mind, I remember we need to inject some kind of uh liquid to
Die.
human. It is PET, right? It's not
Mm.
diffusion MRI. So
Mm.
yeah, I also get this knowledge.
Mm.
All right. If we don't have any more questions or comments, I suppose we should end it here.
Yeah.
Yeah.
Yeah, given that we only have no more than 2 hours to read this paper, I think that's
Yeah.
Wow. That's all we
pretty.
can do. Yeah.
Yeah.
Well, hopefully, the audience is happy with our journal club today, but nonetheless, all right. See you next time.
See you.
Bye bye.